Assalamu alaikum. Today we will talk about globular protein. Globular protein, it's a type of the quaternary structures of proteins with the fibrous protein. We will talk about the fibrous protein in next lecture. Globular protein, amino acid chains folded into a shape that resemble a sphere. Its axial ratio is less than 10. So it's called globular. It's like a sphere. And this type of folding will increase the solubility of the protein in water, as the polar group will be on the outer surface of the protein near water, as it's hydrophilic, and hydrophobic non-polar group will be interior in the interior side of the molecule. We said that before in folding of the protein. You remember this? Hemoproteins are conjugated proteins. What the meaning of conjugated means? That it has a protein part and has non-protein part, so it's conjugated. Its protein part called globin, and non-protein part is the heme prosthetic group, and they are vital for human body. They have heme as a prosthetic group, which is tightly bounded in their structure to the protein part, which is called globin. In hemoglobin and myoglobin, the protein part called globin. Hemoproteins are globular proteins. They are all have spherical shape and axial ratio less than 10. Examples of heme containing proteins and enzymes. There are some heme containing proteins which include hemoglobin, which serve to reversibly bind to oxygen, myoglobin, which contain heme group as hemoglobin, and cytochromes. They are the enzymes of the electron chain, transported chain. They contain heme as electron carrier. We will study it and we will explain it in the biological oxidation in the electron transport chain. Heme containing enzymes including catalase, and the heme part is a part of its active site to which catalyze breakdown of hydrogen peroxidase. Also peroxidase and tryptophan dioxygenase, it's one of enzymes included in the metabolism of tryptophan amino acid. Oxygen binding proteins, which are hemoglobin and myoglobin, they are hemoproteins, and all hemoproteins are globular proteins. Oxygen binding proteins are myoglobin and hemoglobin, and they are the most abundant hemoproteins in human body. We said that the meaning of hemoprotein, they are conjugated protein, have two parts: protein part, which is the globin, and non-protein part, which is the heme prosthetic group. And we will study both parts to know the structure and the function of both myoglobin and hemoglobin. So the non-protein part, which is the heme, is a complex of protoporphyrin 9 and the ferrous iron in the center of the ring. Ferrous iron is held in the center of the heme molecule by bonds to 4 nitrogen of the porphyrin ring, which is the protoporphyrin 9. We will study those ring in details in heme metabolism, and it can form two additional bonds, one on each side of the planar porphyrin ring. One of these positions will be connected to or will be bound to oxygen, and the other will be bound to histidine amino acid, which is present in the globin protein part of hemoglobin or myoglobin. Let's see the figure to understand more the heme structure. Look at this. This is the heme structure, the heme prosthetic group. It's the non-protein part of hemoglobin and myoglobin. It's the same in both. It consists of protoporphyrin 9 ring. This is the ring. 4 biroll rings. We will study it in details in heme metabolism. This is called biroll ring. They are 4 to form the protoporphyrin 9 ring. It has 4 nitrogen atoms, which are connected by bonds to the ferrous iron in the center of the heme group. Also, ferrous iron can form 2 additional bonds at the planar plane of the heme group. This bond to the oxygen and the other bond to the histidine. This histidine is amino acid from the globin part of hemoglobin or myoglobin. Histidine is one amino acid from the polypeptide chain of the globin part of hemoglobin or myoglobin. And this is the heme group, which is present on both hemoglobin and myoglobin. So let's talk about myoglobin structure and location. Myoglobin is one of the heme protein. It's formed of the heme group with the globin part, which is the protein part of myoglobin. It's present, its location in heart and skeletal muscles. It gives them the red color. Their function mainly reservoir for oxygen of the muscle. So it's oxygen reserve mainly. The second function is oxygen carrier, that increase the rate of transport of oxygen within the muscle cells, especially when they are working, exercising. Muscles need oxygen in low concentrations of oxygen. The structure of myoglobin, as we said before, it's formed of heme group plus the globin part, which is a single polypeptide chain, structurally similar to the individual subunit polypeptide chain of hemoglobin. This polypeptide chain is folded into 8 stretches labeled from A to H. Let's see in the figure. So myoglobin structure is formed of 2 parts. The globin part is the protein part, which is the polypeptide chain, and the heme group. Heme group, we explained it before. This is the heme group here in the figure. The heme group, which is formed of protoporphyrin 9 ring. In the center of it, there is the ferrous iron. Ferrous iron will form 4 bonds with nitrogen of the biroll ring and 2 additional bonds, one with oxygen and one with histidine of the polypeptide chain of the globin. It's present here between the stretches F and E. This green polypeptide chain is the globin polypeptide chain, which is folded to form 8 stretches named from A, this is A and B and so on till H. And the heme group present in the center between F and E stretches. And the interior of the this molecule, the myoglobin, contain non-polar amino acids, and the polar amino acids will be present at the external part of the protein, which is near to water, except 2 molecules or 2 moiety of histidine. And the heme group will be located in a pocket in the molecule between the helix E and F. It's usually lined with non-polar amino acids with exception of 2 histidine amino acids. This is the structure of myoglobin. The heme group, protoporphyrin 9 ring with a ferrous iron connected to 4 bonds to nitrogen, 1 bond to oxygen, which will be carried by myoglobin, and 1 bond to histidine. Histidine come from the globin part, here the globin part. And the globin part is a polypeptide chain, single polypeptide chain in myoglobin, only single chain, which folded into 8 stretches. And between F and E helix, there is the heme group, surrounded by non-polar amino acids, except 2 histidine amino acids. One of them is called proximal histidine, which is F8, and it binds directly to the ferrous iron of the heme. And the second histidine is called distal histidine. It's the E7, and it doesn't directly interact with heme group, but help to stabilize the binding of oxygen to the ferrous iron. Here, this is the F helix. Look at this figure. And this is the E helix. Between them in a pocket, there is the heme group with ferrous iron in the center, have 4 bonds to nitrogen, 1 bond to oxygen, and 1 bond directly to the proximal histidine, F8. And this is the distal histidine. Distal histidine is part of the E helix and not directly bound to the iron of the heme group, but it stabilizes binding of oxygen to the heme group. And this is the globin part, the polypeptide chain of myoglobin, which is folded into 8 stretches from A to H. And it prevents oxidation of the ferrous iron of heme, protected from oxidation. What about structure of hemoglobin? There is many types of hemoglobin in our body, but the most abundant type of hemoglobin in the adult blood is the adult hemoglobin A1, and we will talk about its structure. At the end of the lecture, we will have other types and we will explain other types of hemoglobin. So the most abundant type of hemoglobin is hemoglobin A1. We will talk about its location, function, and structure. Its location, it's present in RBCs. It's one of the hemoprotein, conjugated protein, which contain heme group and globin protein part, and its function is to carry oxygen binding proteins. They carry oxygen, so its main function is to transport oxygen mainly from the lung to the tissue, and transport hydrogen ions and carbon dioxide from the tissue to the lung, and we will explain this in details, and also act as RBC's buffer. We will study buffers in details in physiology. Structure of hemoglobin, as we said before, hemoglobin has non-protein part, which is the heme group, and protein part, the globin. But hemoglobin, the difference between hemoglobin and myoglobin, that hemoglobin is consisted or composed of 4 polypeptide chain, not a single polypeptide chain. Myoglobin is single polypeptide chain, but each polypeptide chain is identical to the polypeptide chain of myoglobin, which is folded into 8 stretches of the alpha helical structure, with a heme binding pocket in each polypeptide chain. So hemoglobin have 4 heme group, as it's composed of 4 polypeptide chain. Each polypeptide chain form 8 stretches with heme group in a pocket. And adult hemoglobin A1 composed of 2 alpha chain and 2 beta chains, which are arranged into 2 dimers held together by non-covalent bonds. And the oxygen binding property of hemoglobin are regulated by interactions with allosteric effectors. We will explain what the meaning of allosteric effectors. So again, look at the figure, the structure of hemoglobin. This is hemoglobin molecule. This is the quaternary structure of hemoglobin. It's an example of the polymer. It's formed of 4 subunits. Each subunit is a polypeptide chain, which is similar to the polypeptide chain in myoglobin. It's folded into 8 alpha helices from A to H, and it's formed of 2 alpha chains and 2 beta chains. This is the adult hemoglobin A1. And there is a pocket for the heme group in each polypeptide chain, so it contains 4 heme group. 4 heme group in the 4 polypeptide chains. And alpha chain with beta chain, this will form a dimer of alpha beta, and this is the other dimer, alpha beta 2. This is alpha beta 1 and alpha beta 2. Alpha chain is of 141 amino acid, beta chain 146 amino acid, and this is the heme group in each polypeptide chain. In the center of it, formed of protoporphyrin 9 and ferrous iron in the center. Both dimers of alpha beta dimer 1 and alpha beta dimer 2 will be connected by non-covalent bonds, like this figure, hydrophobic bonds. So the quaternary structure of hemoglobin Hemoglobin is a tetramer. Tetra means 4. Means 4 polypeptide chain, 2 alpha and 2 beta. This will form 2 identical dimers, alpha beta 1 and alpha beta 2, like this figure. Alpha beta 1 and alpha beta 2 connected by hydrophobic bonds. And the 2 polypeptide chains within each dimer are held tightly together by hydrophobic bonds or interactions. In contrast, the 2 dimers are able to move with respect to each other, being held together by polar bond. Polar means ionic bonds and hydrogen bonds. We explained before what the meaning of ionic bonds and what the meaning of hydrogen bonds and what the meaning of hydrophobic bonds. This is in protein conformation. So in this figure, again, the quaternary structure of hemoglobin, 4 polypeptide chain into 2 dimers, 2 identical dimers. Alpha beta dimer, which is connected by hydrophobic bonds, and another dimer of alpha beta chains, and both dimers are connected here by polar bonds, ionic and hydrogen bonds. These are weak bonds that help sliding of both dimers together, and this will change the position of both dimers in the condition of binding to oxygen and the condition of unbinding with oxygen. So the weaker interaction between these mobile dimers result in the 2 dimers occupying different relative position on both deoxyhemoglobin, deoxy means without oxygen, and oxyhemoglobin, it's hemoglobin with oxygen. Here, oxygen binding to hemoglobin. Let's see the 2 forms of oxygen. It's here deoxyhemoglobin means hemoglobin without oxygen, not bounded to oxygen. It's called also taut hemoglobin or T form of hemoglobin. Here are the strong interaction between the 2 alpha and beta chains of each dimer. They are strong interactions, hydrophobic connections or hydrophobic bonds. And here are the weak ionic and hydrogen bonds between the 2 dimers. These weak bonds can be broken easily with the entrance of oxygen. When oxygen comes to be bounded to hemoglobin, this will break the weak ionic and hydrogen bonds between the 2 dimers. So 2 dimers will be relaxed or stretched, and this is what's called oxyhemoglobin. It means hemoglobin with oxygen, or the relaxed form of hemoglobin, or R form. Again, there is 2 forms of hemoglobin: deoxyhemoglobin or T form, or taut or tense hemoglobin. Tense mean compacted with each other, so they are connected. So there is here many weak ionic and hydrogen bonds between the 2 dimers of hemoglobin. When it's bounded to oxygen, this will break the ionic and hydrogen bonds between the 2 dimers, and this molecule will be oxyhemoglobin will be relaxed or R form. This is the difference between them. So there are two forms of hemoglobin: T form, taut or tense, in which the two alpha beta dimers interact with a network of ionic and hydrogen bonds that constrains or limit the movement of the polypeptide chains. And this form have low oxygen affinity of hemoglobin. It's deoxyhemoglobin. The R form or relaxed form, it's the oxyhemoglobin. It has high oxygen affinity form of hemoglobin, in which binding of oxygen to hemoglobin will cause rupture of some ionic and hydrogen bonds between alpha and beta dimers and lead to the structure which is relaxed. So it's called relaxed or R form. Binding of oxygen to myoglobin and hemoglobin. They are both oxygen binding protein. They can carry oxygen. Myoglobin is a single polypeptide chain, so it can reversibly bind to one molecule of oxygen, as it contains one heme group, as we said before. Hemoglobin can bind to 4 oxygen molecule, as it contains 4 heme group in the 4 polypeptide chain. In hemoglobin, there is positive cooperativity. It cooperatively, when the first oxygen bind to the first heme group, this will increase the oxygen affinity of the remaining heme group of the same molecule of the hemoglobin to bind with oxygen. So when hemoglobin bounded to oxygen molecule, this will increase the affinity of this molecule of hemoglobin to bind with the third oxygen, and so on. The third oxygen molecule, when bounded to hemoglobin, it will increase its affinity to have or to be bounded with the fourth oxygen molecule. So the first oxygen molecule is the most difficult, and the fourth have the maximum affinity for oxygen. This is what called positive cooperativity of hemoglobin molecule, that binding of one molecule of oxygen to the hemoglobin in one heme group will increase the affinity of other heme group of the same molecule of hemoglobin to bound with more oxygen and more oxygen. And degree of saturation of these oxygen binding site in all myoglobin or hemoglobin molecules can vary from 0 to 100%, which means full saturation of all molecules with oxygen.